The overall goal of the following experiment is to show the usefulness of a new concept called chips using only visible light, oxygen, and catalysis to functionalize carbon-hydrogen bonds via intermediate hydroperoxides. This is achieved by irradiating a solution of a tetrahydrocarbazole with visible light in the presence of catalytic amounts of a photoinitiator to form a hydroperoxide by reaction with singlet oxygen. As a second step, the hydroperoxide is treated with catalytic amounts of an acid, which facilitates the substitution of the hydroperoxide group by an added nucleophile. Next, chromatography of the reaction mixture is needed, or, as is often the case, simply filtration of the precipitate in order to isolate the pure product. The results show how pharmaceutically active compounds can be synthesized by the concept of chips requiring only catalysts, visible light, and oxygen.
The main advantage of this technique over existing methods requiring expensive metal catalysts or stoichiometric amounts of synthetic oxidants is that we use environmentally friendly oxygen and visible light.
Although we never experienced any problems with these compounds, don't forget that peroxides are potentially explosive. Take precautions, for example, never heat the neat peroxides or never expose them to pure acid catalysts.
Prior to starting this procedure, purify the desired tetrahydrocarbazole if it is colored using recrystallization or column chromatography to obtain a colorless starting material. Weigh out 1 gram of the tetrahydrocarbazole into a 250 milliliter flask. Then add 100 milliliters of toluene to the flask.